Click for IB Biology. Here we're taking a look at gene expression in this particular topic. There's a blank PDF of this presentation down in the description. You can annotate that with your own notes. Take the quick quiz, 10 questions with answers, and there's even a channel link so that you can paste in an AI prompt and have your own individualized self-assessment multiple choice style. So we're just going to take an overview of the concept of gene expression. So gene expression means to switch a gene on. The gene is switched on and it will be producing ultimately a polypeptide or possibly one of the forms of tRNA or rRNA. No cell expresses all of its genes. Of course, all cells contain all of the genes for an organism, but we only switch on particular ones to coordinate the specialization and differentiation of that cell to a particular function. So we only turn on a small subset of the total number of genes within any one cell. The control of expression that we're going to be looking at in transcription, where we go from DNA into the messenger RNA, there is the presence of transcription factors, proteins, which are either accelerating the promoter or suppressing the promoter to begin the work of RNA polymerase accessing the promoter zone and starting the transcription of the gene. So there's a multitude of transcription factors from the environment of the cell interacting with the promoters, turning genes on, turning genes off, and we took a look at that in protein synthesis video 6. Another way in which the expression of the gene can be affected is at the translational stage, where we have the capacity to, once the protein has been made, we can terminate this protein synthesis by breaking down the messenger RNA with nucleases. So that's another way in which protein synthesis could be regulated. So what we've got so far is turning the gene on and off. Number 2 is turning on and off, if you like, the messenger RNA by breaking it down with using nucleases. A third way in which the proteins from gene expression can be regulated is that once they become damaged and or their function is no longer required, they can be targeted for destruction by proteasomes here, and that is achieved by attaching a little protein molecule called a ubiquitin. This targets it and sends it to the proteasome, which digests the protein down to the amino acids. A bit of recycling going on there as well. And a fourth way in which gene expression can be regulated is what's called epigenetics, in which we will study how the environment can influence the expressions of genes, and in certain cases how this can actually be inherited as well. The study of these features of the control of gene expression has, in the 21st century, led to a far greater significance for RNA in what is called the transcriptome, as opposed to the genome of DNA. So the transcriptome of RNA has taken RNA from relative obscurity as a minor player in the genetic story to a major player in the genetic story, such that RNA has even become a major tool of biotechnology. The proteome, the actual proteins that we get, we've seen can be modified proteins through splicing and phosphorylation. Means that the number of proteins that we have totally operating within a cell is far greater than the number of genes available. And so this has revolutionized in the 21st century our understanding of the regulation and the nature of organisms. And we don't make too many recommendations of textbooks at Click for Biology, but this one is a great book, recently published by the Nobel Prize winner Thomas Cech, in which he talks about the role of RNA in the story of life, and how, rather than being a bit part, it is a major player in the actual nature of the functionality of the cell and indeed of the organism. I thoroughly recommend this to either your school librarian or indeed to yourself. Links to the channel are appearing. The next video link is there. There's the channel subscription and the playlist as always.